Hello everyone, my name is Ting Zeng, a doctoral student at Southern Medical University. Is currently mainly engaging in liver disease and depression research with integrated Chinese and Western medicine. Today, my topic of discussion is Indoleamine 2,3 dioxygenase 1 enhances hepatocytes ferroptosis in acute immune hepatitis associated with exercise nitrative stress. Acute immune hepatitis is characterized by the activation of T lymphocytes and the release of a large number of cytokines. Ferroptosis is a recently recognized form of regulated cell death that is characterized by lipid peroxidation. However, the molecular mechanisms of ferroptosis in acute immune hepatitis are largely unknown. In this study, we investigated the classical ferroptotic events in the livers of mice with cacainovaline A to induce AIH. We observed that ferroptosis contributed to the CoA-induced hepatic damage, which was confirmed by the therapeutic effects of ferroptosis inhibitor. Ferroptosis was observed in CoA-induced AIH and contributed to the outcome of hepatic damage. Inhibition of ferroptosis by 4-1 prevented against CoA-induced liver injury by activating system XC. Meanwhile, inhibition of ferroptosis by 4-1 prevented against ferroptosis in human hepatocyte cell line. The dramatically upregulated gene Indoleamine 2,3 dioxygenase 1 IDO1 was identified with AIH, and its role in generation of ferroptosis and reactive nitrogen species was assessed both in vitro and in vivo by genetic deletion or pharmacologic inhibition of IDO1. It is noteworthy that upregulation of hepatic IDO1 and nitrative stress in CoA-induced hepatic damage were also remarkably inhibited by the ferroptosis abolishment. So, we suspected that elevated IDO1 might be contributed to the ferroptotic outcome of CoA-induced AIH. 4-1 could replace elastin-induced nitrogen and oxygen stress in LO2 cell. Thus, inhibition of ferroptosis prevents acute liver injury by alleviating nitrative stress and IDO1 expression. Meanwhile, IDO inhibitor 1-methyltryptophan alleviated murine liver damage with the reduction of inducible nitric oxide synthase M3 nitrotyrosine expression. This indicated that IDO1 is involved in the regulation of acute hepatic immune responses. Additionally, IDO1 deficiency contributed to ferroptosis resistance by activating cellular family 7-member 11 expression, accompanied with the reductions of murine liver lesions and reactive nitrogen species. The absence of IDO1 attenuates immune cell infiltration and inflammation. Consistent with the results in vivo, hepatocyte-specific knockdown of IDO1 led to ferroptosis resistance upon exposure to ferroptosis-inducing compound, erestin, in vitro, whereas IDO1 overexpression aggravated the classical ferroptotic events and the reactive nitrogen species stress. IDO1 aggravated erestin-induced hepatocytes ferroptosis by inhibiting acute in vitro. Thus, IDO1 was necessary for the elevation of nitrative stress induced by erestin in vitro. Overall, our study indicated that IDO1 serves as a key regulator that triggers RNA stress, promoted ferritin degradation, and in turn leading to ferroptosis. These results reveal a novel molecular mechanism of ferroptosis with the key feature of nitrative stress in CoA-induced liver injury, and identify IDO1-dependent ferroptosis as a potential target for the treatment of AIH. Understanding the molecular mechanisms and signaling pathway of ferroptosis may provide a new diagnostic and therapeutic approaches to regulate hepatocytes survival and death in AIH. This is end of my sharing. Thank you.